Welcome to the deep dive. Today we're looking into, well, a really critical part of our immune system, regulatory T cells. You might hear them called Tregs.
That's right. Think of them as the immune system's own self-check. If you have fighter T cells acting like attack dogs.
Then Tregs are the referees, the ones stopping the fight before things get out of hand.
Exactly. They're a specific group, a subpopulation of T cells, and their whole job really is to maintain immune balance, what we call tolerance, prevent autoimmunity.
So our mission today is to sort of unpack the biology behind these cells. They used to be called suppressor T cells, which is pretty descriptive.
It is. Their core function is immunosuppression, turning down the immune response. And we identify them using key markers like CD4, FOXP3, and CD25, especially FOXP3, that's the master switch.
Right, FOXP3, and without it, disaster.
Absolute disaster. If that FOXP3 gene is faulty, you get a condition called IPEX syndrome. It's this devastating, usually fatal autoimmune disease because the body just attacks itself relentlessly.
Wow, so their presence is literally life or death. They have to distinguish self from non-self. How does the body even pick which T cells get this vital referee job? I heard it described as a Goldilocks selection.
That's a perfect analogy, actually. It happens in the thymus, where T cells mature. They're all tested against our own body's proteins, self-MHC complexes.
Okay.
Now, if a T cell reacts too strongly to self, it's dangerous. So it gets eliminated. Apoptosis.
Makes sense. Delete the potential traitors.
Yeah, exactly. If it barely reacts at all, it's allowed to live, but it becomes a conventional T cell, one of the fighters.
Okay, so not too strong, not too weak.
But the ones that react with an intermediate strength, the just right signal, those are the ones selected to become Tregs.
Huh. So the very cells the body trusts to police the system are the ones that were, borderline reactive to begin with. That's quite clever.
It's a fascinating strategy for self-preservation.
Mm-hmm.
So once they're selected, how do they actually do the suppressing? How do they stop the attack?
Yeah, what are the mechanisms?
Well, there are a few ways. They produce inhibitory signals, cytokines like IL-10 and TGF-beta, which basically tell other immune cells to calm down.
Okay, chemical signals.
Right. But arguably, one of the most crucial mechanisms is metabolic. It's about resources.
Resources? How does that work?
Tregs have a huge amount of a particular receptor on their surface, the IL-2 receptor, specifically the CD25 part, which binds IL-2 very strongly.
And IL-2 is important.
Oh, absolutely. Interleukin-2, IL-2. It's like essential fuel for effector T cells. They need it to divide and survive to keep up the fight.
Ah, I think I see where this is going.
Exactly. Tregs act like sponges. They express so much of this high-affinity IL-2 receptor that they just soak up all the available IL-2 in the environment.
Leaving none for the attackers.
Precisely. They effectively starve the effector T cells of the fuel they need to proliferate and sustain their attack. It's a very efficient way to shut things down locally.
Resource control.
Oh, yeah.
Okay, so moving on a bit. I understand there are different types of Tregs. Not all are created equal.
That's right. Broadly, we talk about two main populations. There are the natural Tregs, or N-Tregs. These are the ones generated in the thymus through that Goldilocks process we just discussed.
And their job.
Their main job is tolerance to self-antigens, preventing autoimmunity. They circulate in the blood and lymph nodes.
Okay. And the other type?
The other type are induced or peripheral Tregs, sometimes called I-Tregs or P-Tregs. These develop outside the thymus later on in peripheral tissues. Think barrier sites, the gut, the lungs, the skin.
Ah, places dealing with the outside world.
Exactly. Their job is more about preventing unnecessary inflammation against harmless foreign things. Food antigens, commensal bacteria, that sort of thing. Keeping the peace at the borders.
Okay, that distinction seems really important, especially when we start thinking about disease, like cancer. Tregs often show up in tumors, right?
They do, very often. They get recruited into the tumor microenvironment, the TME. And usually high numbers of Tregs in a tumor are linked to a poor prognosis for the patient.
Because they suppress the immune cells that should be attacking the cancer?
That's the typical understanding, yes. They put the brakes on the anti-tumor immune response, protecting the cancer cells. And that's why a lot of cancer immunotherapies actually aim to get rid of Tregs or block their function. Things like anti-CTLA-4 antibodies partly work this way.
Okay, so Tregs equal bad in cancer, but it's not always that simple, is it? I read something about colorectal cancer.
Ah, yes. That's the twist. In some cancers, like colorectal carcinoma, studies have found that higher Treg numbers are actually associated with a better prognosis.
Wait, how does that work? Why the contradiction?
It really highlights how context-dependent their function is. The thinking is that in certain very inflammatory tumors, The Tregs might not be primarily suppressing the anti-cancer cells. Instead, they might be suppressing the general inflammation in the tumor environment.
And chronic inflammation can drive cancer growth, right?
Exactly. So in those cases, by damping down that pro-tumor inflammation, the Tregs might indirectly be slowing down the cancer's progression or spread. It depends entirely on the specific environment and what kind of fire they're trying to put out.
Fascinating. So modulating them is key. You mentioned depleting them in cancer, but what about boosting them?
Absolutely. For autoimmune diseases or conditions involving excessive inflammation, like maybe even neuroinflammation and ALS, the goal is the opposite. Researchers are working on ways to expand a patient's own Tregs, autologous Tregs, and give them back to restore tolerance.
So manipulating these cells holds huge therapeutic potential, going both ways.
Definitely. It's a major focus.
So as a final thought for our listeners, let's go back to those induced Tregs, the ones in the gut that deal with external stuff. You mentioned commensal bacteria.
Yes, there are specific subsets there, like OR-gamete-positive Tregs, whose development and function are heavily influenced by signals from our gut microbiome.
Like metabolites from fiber?
Precisely. Things like short-chain fatty acids, derived from dietary fiber breakdown by gut bacteria, are crucial signals for these Tregs. They're essential for preventing things like inflammatory bowel disease and even food allergies.
So the microbes in our gut are essentially training our immune system's referees.
In a way, yes. It suggests this whole area of microbially induced tolerance. If we can understand these specific Treg subsets and how they're controlled by external factors like diet and microbes, well, what else could we potentially teach our immune system to tolerate or perhaps react less violently towards? It opens up really exciting possibilities for tailored therapies in the future.